Hello and welcome to Taconic's webinar entitled Genetic Background Characterization and Stabilization in Genetically Modified Models Using Single Nucleotide Polymorphisms and Speed Congenics, which will be presented by Dr. Ana Perez, the head of Taconic's Genetic Department. Please allow me to introduce myself. My name is Margit Wissenbach and I will be your moderator today. Before we get started, I'd like to address a few practical matters. This webinar will be made available to all attendees. We will email a link to the presentation to you as soon as possible. Directly after the presentation, Dr. Perez will answer your questions as they appear in the control panel. So if you have a question, please type it in the control panel. And don't be shy, any question is a good question. The other attendees will not be able to see your question as it is sent only directly to Dr. Perez and me. If you cannot see the control panel, just single-click on the single arrow on the top of your screen. In case your question is not answered at the end of today's session, Dr. Perez will respond to you within the next 24 hours via email. If you're experiencing technical difficulties, please send the host a chat message, and our staff will be able to assist you. So let's get started. We hope you will enjoy today's webinar about genetic background characterization of genetically modified models, single nucleotide polymorphisms, and speed congenics. Dr. Perez holds a PhD in microbiology from Columbia University, New York. In the past, she has been research assistant professor at the University of New Mexico and held a position as director of the mouse transgenic tissue culture histology and microinjection facilities at the Center for Functional Genomics, SUNY at Albany, New York. At Taconic, Dr. Perez is the head of the genetics department and is responsible for the genetic monitoring program and breeding strategies to ensure genetic quality of Taconic's commercial and custom bred lines. In this webinar, Dr. Perez will address the significance of the genetic background for the phenotype of genetically modified mouse lines. Examples of phenotypic variation due to strain backgrounds will be presented, as well as substrain background differences. Single nucleotide polymorphism analysis will be presented as a way to characterize and stabilize genetic background, and as a way to efficiently transfer a mutation to any background strain by using speed congenics. And with this introduction, I would like to hand over the word to Dr. Perez. Please, Dr. Perez.
Thank you, Margit, and thank you, and hello, everybody, and welcome to this webinar. I am hoping that with this I can address questions that you might have, and you can certainly put in practice in your particular universities. So let me just get started here. I cannot get to the next slide. Okay, so during this talk, I'm going to address the following things. This is how am I going to go through the presentation. So we're going to try to understand what is genetic background. We're going to I'm going to give an example of how the phenotype, varies depending on the genetic background. How can we stabilize the genetic background when we have mixed backgrounds, for example? What are single nucleotide polymorphisms, and what are speed congenics? And please do interrupt me if you have questions so that we can address something if you are just stuck with some, any particular pressing issue. So here we have a few mouse showing up here, and I want to just address what is genetic background. We hear about it, but sometimes there's no clarity on it. So genetic background are all the genes and all of the alleles that are present in a particular strain or stock of a mouse, so all of, or a rat, or any being, and it does not particularly include, if we have a genetically modified model, the mutation per se. However, all the other genes that are in that being, in a mouse, being a rat, a mouse, a human, would be what is called the genetic background of that particular strain or stock. So just to have a clarification, what is a homogeneous genetic background? And usually when we talk about a homogeneous genetic background, we're talking about an inbred mouse. And the reason for that is because of the way that an inbred mouse is generated. As a definition, an inbred mouse is a brother by sister mating for 20 generations. So if we carry on a brother by sister mating, we're basically inbreeding. And under those circumstances, at any one locus, we're going to have a homozygous alleles, once we do the 20 generations or so. So, that is by definition what an inbred mouse is. Therefore, because all of the genes, all of the alleles of a particular gene will be in the homozygous state, we will not have any segregation because they are exactly the same alleles for every particular gene. And, each of the strains are multiple copies of a single individual. They are not clones, but they are just, they have the exact background, and they are in a basis, reproducibility of a particular individual. So therefore, that's why it's important and it's useful to have an inbred strain. And we have, many different inbred strains that are commercially available. We have the famous Black 6, FVB, 129, and so we have many inbred strains that have been maintained in this particular way. So as a counter, here we have an example of inbred genetic background, and I just want to give it as a visual perspective so that we, it gives us some clarity on it. We have the three mice that I showed at the beginning. One is a Black 6 and tag, then the other one is a DBA2J bomb tag, 139S6, that these are sold. And now I'm just going to give the examples of these code color genes. These are only five code color genes that I'm citing here. There are many code color genes, but these are the main classical discovered, and just so that we can have a little bit of perspective of what's genetic background. So in this particular mouse, in the black mouse, we have, A codes for the agouti locus, which is in chromosome 2, and it gives us a difference between the black code color or the agouti code color or the, black, and the agouti is really a black and yellow banded hair. So therefore, that's why we have the agouti code color. Then we have the brown locus, which is the TYRP1 gene, as in chromosome 4. And we have a B would be a black mouse, and the B short B will give us a brown pigment. So these are the different alleles for each particular gene here. We have the tyrosinase, famous tyrosinase, which is give us, as is commonly known as the albino locus, is in chromosome 7, and this gives us, the allele of pigment versus non-pigment. Therefore, we would have an albino mouse. Then we have the dilute locus, which is the MYO5A in chromosome 9, and gives us intensity on the particular code color. And we have also the pink eye dilution locus in chromosome 7, and it's involved with eye color. So we have all of these genes, and in every particular strain, and these are all inbred strains, we have a variation of them. And as I said, because they are inbred, in every single example here is a homozygous. So each of these is coding for a particular allele present in this. So, this will be the agouti locus, the low case, the non-agouti here. Here will be the brown locus as a B, and therefore we have the black and the wild-type albino, and so forth. So each particular, you can see that there are different combinations of allele, but in each case it's homozygous. And that combination gives us, a final code color there. So now, as a counterpart, I want to also talk about when we talk about a heterogeneous genetic background, which are in many cases outbred stocks or mixed backgrounds. So, we do sell also commercially available. You have outbred stocks in which are bred in a different way as inbreds, because the goal of these outbred stocks or heterogeneous genetic background is to have a lot of diversity and heterozygosity in all of the alleles. There are different purposes for this particular background, and these mice usually are very, they have more weight than inbred strains. They are healthier. They are easy to maintain. There is allele segregation because obviously there's heterozygosity in different alleles, and therefore, there would be such segregation, and then they would need to be bred in such a way so that we can maximize the level of heterozygosity in this particular line. So, in some cases that we're going to see, we do need mixed genetic backgrounds because in some cases some knockouts are or transgenes could be lethal in a particular genetic background, and then they are only able to be sustained in a mixed background in some cases. So, this is the difference between an inbred and an outbred stock or heterogeneous genetic background. So now I want to just touch base with you guys. What is phenotype? So phenotype is the result obtained from the simultaneous expression of all of those genes from the particular strain or stock. It could be observable by the naked eye, or you can detect it by biochemical reactions. So that's, what would be the result of the genotype. And so that we can see here, the phenotype on all these mice is what we observe as the co-color. So we have the genotype here that is outlined in blue. So this is the particular genotype that we discussed in a couple of slides before. And the phenotype would be black coat color. So even though we have all of these genes present for coat color, we have an outcome of black. For the DBA2, we have, all this would be the genotype. We have a dilute brown. Agouti here in this particular mouse. And for the 129S6, we have a phenotype of an agouti mouse. And this one is the white belly allele, so it will be a white belly agouti. So that's, just to put us in frame of what do we have here. So I just want also to address differences in genetic background. So, and this is important from the perspective of when we decide to use a particular, genetic background, we need to know why do we want a particular genetic background. We need to be aware of differences and to know what would be the best one that would accommodate our experiment. So for example, here we have, this is an open field activity test that had been done here at Taconic by Gerald Botha with several strains that were available. And here we're measuring the particular effect. This is a behavioral test in which you put a mouse, in a spot, and the mouse it adventures itself and moves from that spot where you position it, and goes away from it. So we measure the distance between that spot, and you can see that depending upon on what background. So these are just inbred strains, and each of them are given different results. And you can see the variability on the genetic background without any mutations that had been genetically manipulated or anything. So these are the particular differences. These are the extremes. The 12092SV didn't move much away from the spot where they were positioned, and here and at the other extreme, with the NOD, for example, moved away the most. And there you can see that there's also some variability between females and males. So it's not statistically significant in any of between female and males, but among the strains there are differences, and we just need to be aware of them. So now, when we talk about genetically modified models, we know that they can be generated from using different technologies, and we could use either pronuclear injections, embryonic stem cells, and do bihomologous recombinations, introduce the mutation, or nowadays all the synfever proteins. So we can have different ways of introducing and generating a genetically modified model. So it doesn't matter which way we use. I mean, obviously it depends on the goal of the particular experiment that we're trying to achieve. But the point is that usually these genetically modified models are generated in a genetic background, in a particular genetic background, and it's important to have in mind that when we generate a mutation, we need to be savvy about why using one strain or the other. So here I'm going to give an example of a transgene that was introduced. We had acquired this particular model from an investigator, and is the APP3 mutation is generated as a transgene. So just going back here, this is generated by pronuclear injection. So that means that the particular mutation is going to be introduced anywhere in the genome. It's a random process, and it's going to be integrated randomly. And this particular mouse was selected and defined, characterized, and we have here the transgene called APP3. I'm going to go in the characteristics of it, and we have it in two particular backgrounds. So if we put it in a black 6, or if we put it in a 129S6, and I want to go over and share a little bit of that data. So this APP3 transgenic mouse model is a model for Alzheimer's disease, which is a chronic neurodegenerative disorder, and is characterized by progressive cognitive decline and memory loss, as we all know. And it is characterized by accumulation of beta amyloid protein, and is believed to be an important pathological feature. There's room for discussion here, from the neuroscientists, but this is one of the main goals of this particular model. Why was it generated? APP3 is known as the amyloid precursor protein. That's the APP where it comes from, and it contains, in this particular case, the Swedish mutation, which is an allele that would encode for a double amino acid substitution, and would be that has been associated in humans with the heritable susceptibility to Alzheimer's disease. So these scientists, and I'll go back to Karen Xiao, who is the one who developed this particular model, was first identified in humans, and then it was, generated a transgenic mouse model so that it can be seen and study the effects of this amyloid, the protein deposition, and see what is the effect on it. It does not completely recapitulate the human Alzheimer's, but it can be used to investigate specific intracellular processing pathways, for example, and see how can we either, use it to hopefully truncate those depositions, or, avoid deposition of it. So there's many different perspectives on how this model can be used to study Alzheimer's. So on my next slide, so I want to just talk a little bit about the origin of the model. The origin of the APP model it was developed, as I said, by Karen Xiao Ash at the Department of Neurology and Neurosciences, which she was at the University of Minnesota. This construct was created, as I said, by carries the Swedish mutation form, and it has 2 amino acid substitutions. And here are the descriptions of lysine 670 was substituted by asparagine, and methionine 671 by leucine. So those 2 mutations were identified and therefore put into the mouse. The construct is inserted into a hamster prion protein in a cosmid vector, and is in a reading frame that was replacing the variant APP3 open reading frame. The transgene was originally developed in FEBN mice, but they were poor breeders and died prematurely. So therefore, it was decided to microinject this in a B6SJL F2 single cell embryos to produce the first founders for this particular line. So here we're just seeing a little bit of what genetic background, how is it playing a role here. So then Taconic sells this model, and Karen Xiao gave the model so that we're able to give it to the scientific community, and Taconic colony was established by transferring some of these embryos that were generated from breeding a hemizygous transgenic male to a black 6 intact female. The progeny from a male progeny from that were bred to SJL, and therefore the model that we call 1349, this colony is now maintained by breeding hemizygous transgenic male mice with females that are B6SJL F1 tag mice. So this would be a mixed background strain. To generate, I mean, it's an F1, but still, when you breed again, if you breed it for expanding this, you have to have in mind that if you don't breed it, depending on how you breed it, you could have a mixed strain as a result. To generate the other model that we have at Taconic, the 2789, we use the founder line 2576, and these were by cross for 16 generations to 129S6. Taconic received this stock in September of 2006. The mice were derived by embryo transfer and maintained by backcrossing hemizygous mouse to 129SV Taconic. So you can see that here we have the same transgenic mutation in two genetic backgrounds, and therefore there are two different lines of mice. And I just want to mention a little bit about what is the APP3 characteristics and phenotype on this particular disease when we have it in a mouse as a mouse model. It has early and progressive accumulation of the beta amyloid and development of plaques. It does show behavioral deficits that correlate with the degree of amyloid deposition. There is neuritic dystrophy and altered synaptic efficacy of plaque-associated neurons. There is no evidence of neuronal loss. There is expression of inflammatory mediators by plaque-associated microglial cells and astrocytes. There are some gender differences in some aspects of the physiology and behavior. There is an age-correlated elevation in brain levels of ApoE and cholesterol. And there is deposition of amyloid in cerebral blood vessels of the walls. So this is the phenotype, if you wish, of this transgenic mouse model. So just going back at the genetic background difference for the APP3 model, the model, we have, as I said, the model 1349 in a mixed black 6SJL, and we have also the 2789 on an inbred black 129S6 background. We know that in a black 6 background, the mutation is lethal, and we also know that when we tried, the PI tried to generate this in an FEV background, it was also lethal because they didn't survive. So just we can see on these 4 different examples how the different genetic backgrounds somehow alter the phenotype of this model, and how is it allowing the expression of this particular model. In some cases, it doesn't survive. In some cases, it sustains, and you can see the phenotype development. And this is why we need to have in mind that definitely the genetic background plays a role on the phenotype of a particular mutation. So another thing that we have to have in mind, depending upon what genetic background we are using, is, for example, here there's pink-eyed animals that are associated with certain coat colors, and in this particular case, if we're talking about the B6SJL, the SJL strain carries the PD6B retinal degeneration mutation that causes light sensitivity or blindness in some animals, when it is as a homozygous. So this would impact, for example, if you're doing behavioral studies on how which particular tests you're doing that are depend on vision. So this is something to have in our back of our minds to make sure that when we address or when we do this test, is this the genetic background that we need for this particular test? Would it be better, for example, that I have the 129S6 in this particular, for depending on the study, the focus of the study we're pursuing? So here in the mixed genetic background, as I said, we could have some retinal degeneration depending on, the particular heterozygosity that we have of the mutation that is carried by the SJL. And in the 129S6 model, we would not carry this particular, mutation, because it would be usually in a recessive, but because we're doing, it would be heterozygous, because every time we cross it with 129S6, the particular hemizygote, then if it was there from a previous, if it was carried on into the mutation, then we could wash it out when we're back crossing to 129S6. So all of these things are for us to have in mind when we are working with a model. What are we looking at? What is our background? What are the effects of this background? How is it influencing the expression of my model? So on my next slide, So how do we test for genetic background? So we talk about genetic background, phenotype, genotype, and now that we know and we're aware of genetic background, how do we test for it? How do we keep the genetic quality of the model that we have? So there's different ways to doing this. In the past, there were a lot of biochemical testing done. That was, obviously biochemistry developed, very earlier in life, earlier than genetics, meaning DNA sequencing and the discovery of DNA. So therefore, there are many tests established for biochemical testing of particular proteins that would give us a genetic background information. However, with the discovery of DNA and now more sophisticated technologies, we do have different ways of testing using DNA. When we do biochemical testing, usually we have to sacrifice the animal, and now that we do DNA, we can just take a little biopsy and then with that biopsy either analyze DNA through different technologies that would tell us where we are and what genetic background do we have. So for those that we can do the DNA testing, the major technologies out there are either through detecting DNA microsatellites or SNPs or single nucleotide polymorphisms. So I'm just going to briefly talk about them so that you guys are aware, and forever, if you want to do this in your laboratories, this can certainly be done as controls in your laboratories. So for genetic background tests, DNA microsatellites have been used. What is a microsatellite? Microsatellites are short segments of DNA that have repeated sequence. The length of the sequence repeat varies from species to species or strain to strain, and this is how we differentiate different genetic backgrounds. It is somewhat unstable, and there's a higher mutation rate for microsatellites, so that's something to have in mind. Sometimes there are artifacts because since these are repeats, sometimes they fold, sometimes they can shorten, and therefore that's why we have the higher rate of mutation of them. And it is difficult to perform in a high throughput manner. I mean, as far as I'm concerned, it's been references that the higher level of testing simultaneously of microsatellites has been at the most in, up to 100. I don't think they could do high throughput DNA microsatellites. So that would be, some of the limitations of the microsatellite technology. So then I just want to give an example. This is the way that Taconic used to use and do their genetic background testing for the different strains. We have different strains here, A, J, not D, not SCIDs, and so forth. And in some cases, for example, DBA1 and DBA2 would not be able to distinguish with these two we are testing here for two microsatellite markers. And then when we do, for example, a cross of two different, an F1, which would be two different lines, there you would have obviously 4 of the alleles here, and then you're going to see the different patterns of microsatellites. So by doing this, you are able to distinguish one strain from another. As I said, it's not as high throughput. So nowadays, what Taconic does is we do all of our testing of genetic background by single nucleotide polymorphisms, or SNPs. And what SNPs are just sequence variations that occur when a single nucleotide in a genome is altered. These are more stable than microsatellites and are scalable for high throughput, which is what we like. So we can test in the order of thousands simultaneously. So this is an example of a microsatellite. We have a piece of sequence here. You can see this A. This is the difference, the second base here in this sequence we have for a particular strain an A. And let's say that could be for a black 6, and for a 129, you can have a C. So, many of these, sequences or SNPs can be used as panels if you test several of them simultaneously to be able to design a profile for a particular SNP, and therefore you have a profile of several SNPs for a particular strain, and that's how we test for genetic background. So this is a particular panel that we have at Taconic, 1449 SNP panel that we use from Illumina. And you can see that these are, we're showing here in this particular axis, in the x-axis, the chromosome number. And each of these blue dots is a particular SNP in that particular position. And you see that this particular panel is very nice because it's, you know, it goes very closely. It's 5 centimorgans separate and 5 megabase pairs apart, and therefore you have very nice pattern so that you can be testing all along all of these chromosomes. And this is the length of the particular chromosome. So, you know, they're ordered 1 to 20s, and these are the 6 chromosomes here. But, you know, you can have so many markers here. So there are 1449 SNP markers in this particular SNP panel. And this is just to show how many differences are between those panels. So, for example, if we had a bulb C and tag, if you look here, versus black 6 and tag, so there's a difference of 820. From those 1449 SNPs, there are 120 of those that are you know, different between the 2 strains. So, you know, this is something that, you know, it's nice. You have 120 different markers that you can have the identity of those particular 2 strains, and we can use this for, you know, testing several different strains, as you can see here. There is, I just want to make a point, for example, for the black 6 and tag. If we're testing black 6, there's only 12 difference in this panel between tag and black 6J, for example, which is in the low end. And the reason is that, you know, this particular panel was developed using black 6J as a reference strain, and therefore there are not very many different sequences on these SNPs between the N strain and the J strain. And this, I'll come back a little bit later on that. So just as to finish up my example of the APP3 model, I just want to show you that to make sure that we have a good genetic quality on this transgenic model, it's important to have 2 different checks. First, the genetic background by any of the technologies that we have used, to make sure that we have, you know, a clear picture of where are we with our genetic background, and that indeed, if it is in a, you know, 129A6 background, indeed that's it is in a 129A6 background, or that there is no been any genetic contaminations. Sometimes, you know, there are unwanted and they sneak up on us sometimes, genetic contaminations on mismating usually, and that's something that, you know, we want to make sure that we're checking for. And then obviously genotyping in transgenics or genetically modified models. Obviously, you need to know the genotype of the model. Many times we do set and, you know, have the genotype, and we're doing a home-by-home mating, and we think that everything is bright and perfect, and sometimes we're surprised because something happened along the way, and therefore, you know, you think you have a home-by-home mating, and then, you know, oh no, it turns out that now it's heterozygous. Oh yeah, we didn't test that colony for a long time, and you know, it was a home-by-home. What's the point of, you know, nothing is going to happen? You know, accidents happen, and we need to have that in mind, especially when we see that somehow our, you know, phenotype of the model that we thought we had, you know, it's not the same, you know, and obviously a head from a home is going to have a different phenotype in many cases. Another thing that we have to have in mind with transgenic models, there are some transgenes that are more stable than others. And, you know, transgenes, when they insert in the genome, usually they do in a head-to-tail array, and they could insert as 2 copies or 300 copies. Sometimes there is rearrangement of the transgenes, and we need to make sure that that transgene is still there and has not been rearranged. Sometimes, you know, and this has been published, there are, you know, more unstable transgenes, and the phenotype can be lost. So anyway, so let's go to something else that I want to make you aware. There are some phenotypic differences between substrains, and this is something that I want to press up on you. There are differences between black 6 and tag and black 6J, and I just want to point out here four big differences here. When we did the open field behavioral test in the open field that I showed you in a few slides before, there was a black 6 and tag black 6J difference, significant difference between both of the strains in the open field behavioral test. So this is something to have in mind. Just because we're talking about a black 6 mouse, it doesn't mean that the black 6 and is the same as a black 6J. We need to make sure that there are substrain differences, and we need to be aware of them. There has also been metabolic parameters that are different in some diabetes-induced obesity between BLAC6 NTAG and BLAC6J. So this is something to understand and have in mind. We have, and I'm going to show you either other mutations here that the BLAC6 gene carries. You know, BLAC6 gene has the NNT deletion, BLAC6 NTAG and BLAC6J bomb do not. Then also in the DIO, for example, model, there has the weight gain and adiposity levels are higher in BLAC6 NTAG than in BLAC6J. So these are 4 little isolated examples that I put here to make you aware that substrain differences are also critical sometimes in the model when we're studying our model. So it's important to have this in mind and to make sure that we are, you know, this is in our radar. So let me show you examples of why are these substrain differences in the particular genetic background. As I mentioned before, NNT. What is NNT? NNT is a gene that is the nicotinamide nucleotide transference and is associated with impaired glucose homeostasis and reduced insulin secretion. It is reported as a 17 kilobase per deletion, and it is a gene that is really involved in transferring, reducing the NADP into NADPH. So this is in the mitochondria, and therefore in stress situations, it could affect processes that are involved in stress situations, cancer, stress in the cardiovascular system. And so there's a lot of literature out there, and you can look at it. We have it on our website, so you can look in somehow how has NNT been involved in different processes. There's also this SNCA or synuclein alpha associated protein, which is called the SNCA gene, and gives resistance to the effect of MPTP, which is, I'm not going to say this whole name, but it is really a drug, and it obviously is involved in dopamine level metabolism. It's 365 kilobase per deletion, and if we're doing, for example, behavioral studies, or because this is involved in metabolism in neural cells, obviously this would affect if we're testing a drug that is in that pathway, or we're trying to study something that involves this particular pathway. So we need to be aware of these differences. And finally, there's a recent one that has been lately been in the transgenic list, CRBP1-RD8 mutation, which is a single base per deletion in the CRAMS homolog 1 gene and causes a mild form of retinal degeneration. So why am I talking about this? So I just wanted to show to you that there are differences between the BLAC6 substrain, as I told you before, and that depending on the mutations of these genes, these are genes that have been discovered and have been found in inbred strains without, they have been found in the different strains, and therefore they have an effect. And obviously there are in inbred strains as a process of natural DNA generation and repair. Sometimes, some mutations are not repaired, and they would be, fixed in a particular substrain, especially because they are separated. So we have different flavors of BLAC6 mice. Remember, a BLAC6 mouse is not a BLAC6 just because it's a BLAC6 color. We know that, but also there are substrain differences: the BLAC6J, the BLAC6 NTAG, the BLAC6J bulb, the BLAC6 NCRL, and so forth. So, for example, I told you about these mutations here. Where are they? And they're present. So, for example, NNT is present in BLAC6J. However, BLAC6J does not have the SNCA or the CRB1-RD8 mutation. Here we have another BLAC6J OLA that is sold by HSD, and then it does have the NNT mutation, it does have the SNCA mutation, but does not have the CRB1-RD8 mutation, and so forth. So here BLAC6J bomb does not have any of those mutations. The BLAC6 NTAG carries the RD8 mutation, but does not carry the NNT or the SNCA. And the BLAC6 from Charles River N, the same thing, does not carry SNCA or NNT, but also has the RDA mutation. So anyway, just bringing awareness on there are substrain differences, and we just need to be aware of them. So Taconic, because we have been asked by many scientists, how, now that I know that there's a black 6 substrain differences, how can I test for it? So Taconic has developed a black 6 substrain SNP panel, and this is just like the other SNP panel that I show you. This panel is not as big. It's a 96 SNP panel, and each of the blue dots here represents the markers along the different chromosomes here too. And you can see that in some cases, there's, for example, for chromosome 1, we have only 4 SNPs that are positioned along this chromosome and so forth. So in some cases there are some bigger holes. However, commercially available, this is one of the few black 6 substrain panels that you're able to differentiate between black 6J and black 6N. And here's how, many of the difference that we have, black 6J and black 6N, we have 94 of those SNPs are different between the two of them. And so, it helps us distinguish the black 6 background that we have here. We also have the black 6J bomb from black 6N tag, for example. There's 47 differences here. And so we have comparative SNPs here and there. In some cases, we are not able to differentiate. And the reason for that is that, we need to do more sequencing work to be able to find more differences. But this is useful, definitely, to be able to distinguish black 6J bomb from N tag and J from N tag. So, talking about how do we stabilize this mutation from a mixed genetic background, how do we go about that? And why would we want to do that? So we've seen that in some cases it's important that genetic mutation to be able to stabilize that we have a mixed genetic background. In other cases, if you want to publish your data and you want to publish it and show that you have done a comparative analysis between this mutation and this other mutation, and they're both comparative, you would like to have a homogeneous genetic background. So if you have that particular scenario, how do you transfer your mutation that it was generated in a 129 black 6 background into a particular inbred background? We know that phenotype varies depending on the genetic background. So how do I stabilize my mutation that is in a mixed genetic background to a particular single genetic background? So we do that, as we know, through back-crossing into a particular defined genetic background and develop a congenic strain. So I'm going to address a few of those concepts here, and I'm going to show an example so that you guys, hopefully visually is easier understandable than just talking about it. So, for example, we have a Taconic about nude mouse. It's a cute little nude mouse here, and with the albino gene there. This is the nude mutation. It's in chromosome 11, and we transfer it to a black 6 genetic background. So we took this nude mouse, we back-crossed enough times to black 6 to be able to develop our black 6 nude. And so to do that, the process of doing this back-crossing and transferring the mutation from one inbred strain to another, or from a mixed strain to an inbred strain, is called a development of a congenic strain. And for that, we have two important concepts. The donor strain, which is the original mouse that carries that particular mutation, onto another genetic background, which is going to be called the recipient strain. So for that, we did back-crossing. So what is back-crossing? Back-crossing refers to a mating scheme when an existing mutation in a particular line is purposely mated to a different strain. To be defined as a congenic line, it has to be to an inbred strain. And this is done by consecutive generations of back-crossing. So as I said, there's two components, the donor strain and the recipient strain. The donor strain will be giving you the gene of interest that you want to transfer, and the recipient strain is going to be the strain that is going to be accepting that mutation for that particular genetic background. So when we talk about normal back-crossing, if this is defined and there are, obviously the way that DNA replicates, every time that we replicate, half of that particular genetic background goes to another. So if we had a particular mutation in a background, when we inbreed it for a particular, when we cross it into other particular strain, we know that half of the chromosomes are going to come from one animal and the other half from the other animal. So the two different strains are going to give me the composition of the final mouse here, and therefore we're going to be transferring every time that we do this type of crossing. We're going to be talking about 50% of that donor animal being transferred to the recipient animal. And when we do this enough times, but 10 back crosses, hopefully we transfer our mutation into the new genetic background. So this we do, and as I said, the donor strain would be our nude mouse here, and it's going to be transferred to a black 6. So we go back and forth, we take this strain, we take the result of the cross, and we cross it again to the recipient strain, and we say we do that to have 10 back crosses, we have a full congenic strain. And usually when we do that, and I said here we have the source mutation, and the recipient strain will be the defined genetic background strain that will be receiving the mutation. Obviously, every time we're doing and selecting these pups, we have to genotype to make sure that we follow our mutation. Otherwise, we can lose the mutation, but we want to be able to bring this mutation into the new strain. So every time we back cross, this is just an example for us to get a better picture of how this happens, is, this is an example of a second back cross, okay? So this is the number of animals that we have. This is the percentage of black 6 that we have in each particular bar here. And this is at a second back cross. So here we made it just a pair of mice, and we collected all 33 progeny from, a pair of mice of our donor and recipient, and you can see that there is really a normal distribution in the way that the genetic background is, distributing in this population. And so in this particular case, we have one mouse that was, generated that had 65 to 67% of black 6 background. We had in the middle 10 mice that had 74 to 76% of black 6 background, and at the other end we had 3 mice that have 80 to 82% genetic background, black 6 genetic contribution. So you can see that there's a very big variation. So if we're doing this the classical way, and we do our back crosses, we made it to mice, we go back and we choose one of these paths, we open our cage where all of our mice are, we take a mouse here, the probability of getting one of these 10 mice is very high, so we might get either this or this mouse. So we will have, a contribution for the next back cross of a high probability of having 74 to 76% contribution. Wouldn't it be lovely if we could get this mouse here for our next back cross? So yes, if we do that and we're able to test for the genetic background, we can select our population. And this is what we do, and it's called speed congenics. This is just an example of how when we test for SNPs, how can we choose, and we would know reference markers here, and this is just an example. So if we have homozygous and it correlates with a particular strain that we want, we would have, a particular background and select for the percentage, a higher percentage of genetic background for the next crossing of the next population of mice. So I just want to make sure that we understand, before I go back there, that we have all of these tools to be able to generate mice and select the ideal mouse for the next generation to enrich and get the best probability of getting further there. And before I conclude here, I just want to make sure that if we do, for example, speed congenics, instead of going for N10 generations, we would be going for taking a half of this time, and if we go for speed congenics, we will only need to do 5 back crosses to be able to get to stabilize into a full congenic strain when we are able to test for a particular for the higher percentage of the genetic recipient background that we want. So this can be done by SNP testing or microsatellites, and then we can select and do a very I guess backcross, educated backcross, to be able to pick the best animal for the next generation of backcrossing. So going back to my final talk, we've reviewed as a summary and conclusions what is phenotype and genetic background. The phenotype of a mutation in many cases changes with the genetic background of the strain that is being used. We are aware of substrain differences, and we need to have that in mind when we are using different, strains, inbred strains of mice. And to stabilize a mutation, we need to put it in a defined inbred background if it's able to survive on that particular background. It facilitates interpretation of the data, and if we want to compare on, different publications, if there is some other mutation that we're trying to compare it, we should put them both in the same genetic background. Congenic strains are established by repeated backcrossing to the recipient strain, and there are two methods involved that are available. Traditional backcrossing, in which you're not able to test for the background and you just go for 10 generations, or speed congenics, in which you are able to test for the background and facilitate and speed the process, and that's why we call it speed congenics. I have, seen in many cases that with traditional backcrossing, sometimes even though you've done 10 and even to 12 backcrossings, we don't get, fully congenic. You are, 98%, 99%, but in some cases, not always, but in some cases, since you're not able to test for the genetic background, you don't get fully congenic. And then as a way of using, tested genetic background, SNPs enable identification of the mice with the highest percentage of recipient strain background in each backcross round, and therefore facilitates and speeds the process of getting a congenic strain. I think that's it pretty much. So I think at this point I am pretty much done.
Thank you, Dr. Perez, for a great presentation. I think we immediately go to the question and answer session so we can get a few questions answered. I can see one here, one moment. So a question is it possible for a phenotype to be expressed in only heterozygous mice strains for a gene mutation or knockout, but not in the pure mice line?
Yes.
And
yes.
I think I understand that question as, would there be a phenotype of the mutation in a mixed background versus a pure background? I think that's the question.
Yeah, I think what they're asking is if there could be, if a phenotype might be expressed only in heterozygous mice. So as phenotype
Right.
would vary with the genetic background, and therefore in some cases it might vary depending upon, so if you have an inbred strain, it could be in that particular inbred strain but not in another, or it could be lethal in one and therefore you're not able to see your phenotype because it would be very early lethal, and therefore if you put it in a different strain, you might be able, in a different genetic background strain, you might be able to see what the phenotype is for that particular strain. In many cases, you do have the same phenotype. Sometimes you have very dominant mutations that is very clearly cut. They lack, one kidney, if you wish, or there is some deformity in the limbs. That it is, it could be, in many cases, could be the same, in a particular strain and another. But there are many examples in which not necessarily the phenotype would be the same. So the answer is yes.
Thank you. Another question is, panel for SNP, I understood, are actually the most specific nucleotides on that chromosome in comparison to other strains. So I think the question is how, for the panel that we have, how the SNPs are chosen. That's how I understand that that question.
Yeah. Yeah, I think there, when I mean, and I think when we talk panels, we can choose and we can build different SNP panels for, testing different things. There are many SNPs now with sequencing identified in different strains of mice. and we pick and choose the ones that are ideally you would like to have as many as you can. I mean, obviously, you know, they're expensive. If you test for, you know, 300,000 SNPs or so forth, I mean, it becomes obviously more expensive. So, but you do need to choose. The ideal is to have it nicely spaced so that you're able to test all along the chromosome. And yes, they would be very specific for a particular for a different strain. So the different panels would be characteristics for each particular inbred strain. We have a profile, what I call a profile of SNP panel. And yes, they are stable and therefore specific for a particular strain.
Thank you. Another question is, how do you resolve SNPs which are discordant in a specific region, which seemingly indicate a very short chromosomal crossover?
Okay, yes. So sometimes they are, so if you, let's say your mutation is in a particular area, and this is interesting because sometimes we know that there are different kinds of DNA. Sometimes the DNA is very accessible, and sometimes the DNA is, you know, is not as accessible. And there are obviously some areas that are more recombinogenic than others. So if you have, for example, a SNP marker that is very close to your particular mutation that you're trying to breed into this new inbred strain, you might not be able to ever convert it. In some cases, it's very hard. The majority of the cases you're able to. I have seen a couple of cases in, you know, it's just so tight and it's in this area that you are not able. This is not able to recombine, and it's very close to the mutation. Sometimes it's just so close, and just because of the distance of the mutation to the SNP, you don't have enough DNA there to be able to establish a recombination event. So therefore, you talk about link of the SNP with the mutation, and then you would have to make a decision to just be able to keep that particular marker, and you are aware that, you know, a few hundred base pairs would be from the other background, and you're not able to convert it. So there's always a distance between that in which you are able to have a recombinogenic event that is closer to your mutation.
Okay, thank you. Another question that's a bit longer, so I'll read it up slowly. I have a mutant mice in B6J and 126 het mice. When I try to cross it with a pure B6J or 126, the phenotype for the same genotypes disappears completely. I have to cross the SIPs to get the phenotype, and the pair I have is not a good breeding pair. Why may be the reason, and how to counter it?
Yeah. So yeah. So I mean, in this, I mean, sometimes we have these kind of scenarios, and it is possible that when you have this mixed background, you are, you know, selecting for certain contributions of both of those strains that would allow the expression of this particular phenotype. Now, what I don't know if you can counteract, but something that would be useful to do here is to know what percentages of both of those strains are contributing to your phenotype. And sometimes I've seen some cases in which a balance of the mixed background allows for the expression of the phenotype, and then you would need to characterize this a little bit better and know what percentages of the black 6 and the 129 would give you that particular phenotype. It's a tricky scenario, and it's sometimes tough because with mixed backgrounds, it's sometimes hard to stabilize, you know, the mutation, and you have to be crossing and going back and crossing, and what you should really do is freeze your strain so that you have cryopreservation and of this, and you know, sometimes you can save that and whenever you have that, go on back and test some of it and bring it back, and hopefully you have some of your mutation frozen and you're not able to lose it, because if you don't see it, even though you're testing for it, sometimes it helps to have that assurance that if you have this combination of the particular backgrounds, you would see it. So that would be one thing that you can do. I think it's difficult to be able to clarify that, though.
Yeah, that may be a very good advice. So cryopreserve your line to keep it.
Yeah.
Right. Another question is, which is better, to use male or female of donor strain with female or male recipient strain for developing a congenic strain?
Yeah. That is very important because, and I didn't mention in my talk because it becomes very complex, but it is important to have at least in one cross, you need to have a female donor that carries the mutation with a male from the recipient strain. Because you need to fix the Y chromosome. This is the only way to fix the Y chromosome. Most of the markers out there in SNP panels do not test for the Y chromosome because, there are very few SNPs that have been, defined in the Y chromosome, and therefore, because it's such a short chromosome and usually does not code for many things, so it's difficult. Most people do not go after that. But, one thing that we need to make sure, though, because we're not able to test for the Y chromosome, is to fix the Y chromosome from the recipient strain that you want to do. So you do have to have at least one cross in which you have a female donor strain that carries the mutation to a male, recipient strain. The rest of them could be, because usually you prefer to have male donors because you can have a bunch of females that are plugged with the recipient strain. But do not forget the critical step of using at least one cross and selecting, perhaps from that cross for the next generation with a female donor.
I think I'll take one more question. Our time is a little bit advanced, and please be sure that your questions will be answered anyhow, even though we may not be able to address them. So one more, I think, before we may have to close. What would be your recommended number of animals to send for SNP analysis in order to identify the mice which have the high percentage of recipient genome?
Yeah. So I think, there's been several different publications about it. There's many nice publications that include a lot of mice. I mean, I think a critical number that is talked about is 20. A lot of people find, hard, because obviously when you're breeding this, you have only half of the animals going to carry the mutation, because every time you backcross, you have only heterozygous mice carrying the mutation that you want. So, but I think a safe number is between 8 to 10. That allows you to advance enough and, to get there in 5 generations if you're testing your background. So 8 to 10 is a reasonable number. If you cut a little bit more than that, sometimes you don't have much. But when you go, you shorten yourself because you, are excluding the possibility of having a good recombinatorial event. So, but I think a safe number would be 8 to 10.
Okay. Thank you very much, Dr. Perez, for a wonderful presentation. Thank you for the questions. I think with this, I will end our today's webinar. Thank you for attending, for your interest, and we hope you enjoyed the presentation and the question and answer session and got some inspirations for your own project. We also hope to welcome you again at one of our next webinars, so please keep an eye on our webpage. Our webinars are usually announced on the front page, and we'll hope to welcome you again. So, bye-bye.
Bye-bye.